All right, why don't we go ahead and get started? So the last lecture, when we met, we were talking about situations where the immune system is no longer functional, and we provided some details about how that might come about as a consequence of infection by virus or bacteria or parasites. And as well, towards the second part of that lecture, we also talked about hereditary immunodeficiency. So mutations that exist, for example, in the interferon gamma receptor gene that result in inactivity in that pathway. And so the result of that is that you have these immunodeficiencies. But today we're going to be talking specifically about a virus called HIV, the human immunodeficiency virus, and how that can then lead to acquired immune deficiencies and results in something called AIDS, which is acquired immune deficiency syndrome. So we'll talk about that. We'll talk about the structure of the virus, the HIV virus, as well as some of the host response genes. And we'll also talk about how the HIV virus gets into CD4 T cells. And we've heard about some of these evasion strategies that some viruses use, like the herpes viruses, in trying to prevent an immune response. In this case, by attacking the CD4 T cells, you basically wipe out that critical subpopulation of T cells that are involved in helping dictate the sort of response that needs to take place. And then we'll talk a bit about the host response to HIV infection, as well as some current therapies, and then finally, as a result of long-term infection, how that can then ultimately lead to progression to AIDS. Okay, so what is AIDS? So AIDS is basically a disease that was first described in the early 80s. So this was basically a disease where people that were coming in basically they were sick. They looked like they had something like a mononucleosis or flu-like symptoms. And if they looked at their blood counts for CD4 T cells, they found that there was a massive decrease in the number of these CD4 T cells. But it wasn't really clear what was causing this disease. And it took some time before scientists were able to discover what was causing this. Now, some of these infections resulted in people that were otherwise healthy being unable to deal with essentially benign kinds of infections. So for example, people were starting to develop things like Kaposi sarcoma, B-cell lymphomas, things that, and they were getting sick to things like pneumonia and so forth that would normally be able to be dealt with by a healthy individual. So finally, it was discovered, this HIV virus, human immunodeficiency virus, which is a lentivirus. That's a form of what's called a retrovirus. And a retrovirus is, as we'll see, is something where the viral genome itself is encoded in RNA. That is then integrated into the host genome to make a provirus, and then that provirus then is transcribed to give rise to the RNA virions that then are budding off of the cell. And we'll describe in a bit the life cycle of this HIV virus. Now, it should be noted that obviously this is an important discovery, because once you discover the etiologic agent of a particular disease, then you can start to combat it. And so both Robert Gallo and Luc Montagnier independently discovered, or at least supposedly independently discovered, this virus. And for interesting reasons, Montagnier solely went on to win the Nobel Prize in 2008. So there's a bit of a interesting story about how all this goes, and I'm not going to get into that today. So basically, there are two types of HIV, strain 1, HIV-1, and HIV-2. And HIV-1 is the more virulent strain. It's the one that you tend to find in populations where this disease is common. This is also the principal cause of this acquired immune deficiency syndrome. So this, as we'll see, has an interesting way of dealing with infection and being able to wipe out the immune response. So this is an EM micrograph basically showing the structure of these virions. And you can see this, this little layer here. This is a membrane that's basically derived from the host cell. So once it infects, is replicated inside of that host cell, it will bud off that cell and take with it a piece of that membrane. And associated with the membrane, you see these little spikes. Those little spikes are something called GP120, which is a glycoprotein that's produced by the viral genome and is essentially a transmembrane protein, so it makes its way to the surface of these virions. And as we'll see in a minute, that's involved in the infectivity, the specificity, and the ability of this virus to infect CD4 T cells, cells that express CD4, I should say. So if we look more carefully at the overall structure, the schematic basically shows the overall structure of the virion. So again, here you can see this GP120 protein on the surface, and then associated with that is another glycoprotein called GP41. And these guys, basically their function is to recognize the appropriate host cell, and that host cell, as we'll see, is expressing CD4. And the result of that then is that it can bind to that cell through that receptor and then mediate its way into the cytosol of that cell. And when it does so, it releases this structure called the nucleocapsid. And the nucleocapsid is basically made up of these nucleocapsid proteins and a variety of the viral RNA. So this is the genome of this virus, as well as some of these proteins, these enzymes like reverse transcriptase, which basically takes this messenger RNA and then can convert that into a cDNA, which then becomes integrated into the host genome. As well as a protease that is involved in breaking down, once it enters into the cell, it can then break down some of these polyproteins. Have you guys ever heard this term polyprotein before? So basically, some types of proteins that are encoded by viruses and some bacteria, they start out as a longer protein, and then they're cleaved into smaller parts by an enzyme. And in this case, this occurs through the nucleocapsid protein, has a protease function involved with it. And so the result of that is, for example, there's a polyprotein that is broken down by cleavage, and that will generate this GP41 and GP120 that are both present on the surface of this virion. Okay, and then obviously for infectivity, you need to have the reverse transcriptase, and you need to have some of these other proteins in order to be able to function. So if we look at the different genes and the structure of this genome, so again, this is a viral RNA. It's basically looks something like this, where you have all these different genes. Some of these are encoded as polyproteins, so they need to be cleaved by these peptidases. But basically, if you look at the overall structure, the linear structure of this RNA, at the 5' and 3' ends of this, you have something called an LTR, which is a long terminal repeat. And basically what that LTR is doing is that helps to allow this virus to make its way and to integrate into the host genome. And as well, it's also got a very strong promoter here on the 5' end of this messenger RNA. So the result of that is that under the appropriate conditions, you start to get, once this is integrated into the host genome, this LTR has appropriate DNA binding elements where transcription factors can come in. And the result of that is that you start to get transcription of some of these different genes, and ultimately that then leads to the ability of this virion, or I should say, in this case, it's a provirus, but once it starts to make those genes, then it can start to replicate, make little copies of itself in the cytosol, and then be able to butt off the cell and go on and infect other cells. So some of the different proteins that are involved, I don't want to go through all of these, but just a couple highlight. You have GAG here, which is basically some of these core proteins and the matrix proteins that give rise to the nucleocapsid sequence. You have the polymerase, which is the reverse transcriptase, I should say. And that also is a polyprotein which is broken down by protease, as well as giving rise to these integrase enzymes. And then ONV, which I talked about before. So these are these surface proteins that are on the surface of these virions, and that encodes GP120 as well as this GP41, both of these glycoproteins that are on the surface of the virion, and et cetera. So all these are essentially regulatory proteins that are required for the life cycle of this virus. And they've all evolved, or essentially co-evolved with CD4 cells to be able to get into those cells, essentially develop a state of quiescence, and then ultimately, when those CD4 positive cells become activated, they then can essentially start to replicate again. So they go from a provirus stage, which is integrated into the host genome, and then make it back out and start to make productive virus. So let's talk in more detail about the life cycle of HIV. And in this case, infection is taking place in CD4 positive T cells. So here you have the virion, and on the surface of a T cell you have CD4, this is a helper T cell, It also has a co-receptor, and as we'll see in a minute, these co-receptors are chemokine receptors. We've talked a lot about chemokine receptors, but these guys are obviously involved in getting the T cells to the right place in the body, and in this case, these are typically chemokine receptors that are associated with activated T cells or memory cells. So here the virus comes along, it's got GP120 and GP41 on the surface. That then binds to CD4, and the result of that is that that binding kind of changes allosterically this GP120 structure such that GP41 can now bind, and the two bind to both CD4 and to this co-receptor. So the result of that then is that the virus starts to essentially fuse. Its membrane here, its envelope, starts to fuse with the host membrane, and the result is that this nucleocapsid, which is shown together here, that nucleocapsid starts to make its way inside of the cytosol of this cell. And so that then leads to the breakdown of the nucleocapsid and the release of the viral RNA, as well as some of the other enzymes that are associated with that. And so the consequence of that then is that because you have reverse transcriptase, this little blue dot, it can take that messenger RNA, or that viral RNA, I should say, and start to transcribe cDNA, complementary DNA, which is double-stranded, and then by binding to integrase can then bring this into the nucleus of the cell, and also then direct that piece of cDNA to basically enter into the host DNA. So it just incorporates somewhere inside of the host genome, and it's basically just sitting there, and we call this a provirus because it's no longer a virus anymore, it's just a piece of DNA. It's just a sequence there, and in this case, it's quiescent. So this virion has gotten in the cell, it's infected the cell, but it's not productively generating any new virions. It's not replicating. And I should also note that, this CD4 molecule, we always think about that as a helper T cell. That's usually where we find this expressed. But it is also expressed in other immune cells, and examples of that are on monocytes that oftentimes will convert into macrophages and into dendritic cells as well. So these guys we know can play a role in the overall infectivity, and we'll talk about that a bit later. But the important point is that you need to have CD4 on the surface, and then on top of that, you need to have this co-receptor. And if you have both of those, then the cell is then subject to, or it's infectable. The virus can get into the cells and induce replication. Any questions about the life cycle here? The beginning of the life cycle, I should say. All right. So with time, as I mentioned, when this provirus is established inside the host genome, the result of that is that you have essentially a quiescent infection. So the cell is infected, but it's not actively producing any new virions. So what has to happen? So in a T cell, what happens is that if this infection is taking place in these T cells, you get this low level, you basically get this quiescent provirus. But then if the T cell becomes activated, we've talked about this before when we were talking about T cell activation pathways, some of the transcription factors downstream of the T cell receptor, and one of those is NF-kappa B. So if a T cell, in this case, becomes activated, so if it's a CD4 T cell and it somehow becomes activated, the result of that is that you get induction of NF-kappa B activity. And that NF-kappa B, there's a transcriptional binding site basically in this provirus. And so the result of that is that you start to get these low-level transcripts from this provirus. So the viral RNA starts to be produced, and from that you actually start to now take some of those transcripts and start making some of the early genes that are required for making a virion. So examples of that are these proteins TAT and REV, which basically then get these messenger or these viral RNAs out of the nucleus and back out to the cytosol. And then in addition to that, you also start producing GP160, which can then get converted into these proteins on the surface of the virion. So ultimately then, once these are all present in the cytosol, they can start to generate a nucleocapsid, taking up some of this newly transcribed viral RNA, which is transported out into the cytosol, and then start to bud off from this membrane. So here, again, the host membrane is being used by this virus, basically using those lipids to make this envelope that's around this new virion that's budded off the surface of the cell. And so then these are now infectious virions. They can go off, they can infect other cells, and the whole process can then start over and give rise to infection of different cells. And so ultimately, just like any other virus, you know, the strategy is to try to take advantage of the host mechanisms for replication and translation and so forth in order to make new viruses that can go out and infect other cells. Okay, so I wanted to show, actually before I show this movie, do we have any quick questions about the life cycle? Yeah, over here.
I don't know if this is off topic, but I understand that HIV doesn't exist very well outside the blood.
HIV does not exist very well outside the blood, yeah.
Is there any molecular reason for that?
Yeah, it's a good question. So the question was, why is it that HIV is known to be labile in the environment? You know, you don't just get it like you would get the flu bug or something like that. A lot of that has to do with the fact that it's an enveloped virus, and so these lipids are not particularly stable in the environment. They need to have, you know, they need to be basically in blood in order to stabilize them. Any other questions? Way back there.
So is GP160 like a polyprotein that leads to the other?
Correct. It's cleaved by protease to give rise to 120 and the 41. Any other questions? Yeah.
Any other host cells that they can use to do the transcription and reproducing themselves rather than the CD8 T cells?
Are there any other host cells where you can get replication? Yes. So they can replicate in dendritic cells and macrophages. Although most of the time this is going to be taking place in T cells but those other cells, they also have activation of kappa B through the toll-like receptors, for example. Right. But it's primarily, most of the infection is actually going to take place in these CD4 cells because they just do a really good getting into them. Okay, let's take we'll take an opportunity, because it's Monday afternoon, I guess, to watch a little movie.
In its efforts to combat HIV, the pharmaceutical community has in its crosshairs what a field general would refer to as a target-rich environment. In its first brush with a T cell, the HIV particle uses its GP120 surface glycoprotein to snag a CD4 membrane protein and its co-receptor, triggering the harpoon-like GP41 glycoprotein core that draws together and fuses the viral and host cell membranes, propelling the viral core into the T cell cytoplasm. An uncoating process releases the viral capsid containing 2 copies of viral RNA, associated proteins, and the viral enzymes vital to replication, reverse transcriptase and integrase. Once in the cytoplasm, reverse transcriptase creates a double helix DNA copy of the virus RNA that is circularized and covered on its ends with a pre-integration complex of proteins, including a tetramer of integrase molecules. Coordinating magnesium ions at their active sites help to hydrolyze a dinucleotide from each 3' end, exposing new hydroxyl groups. The pre-integration complex guides the proviral DNA to a host nuclear pore and threads inside to find a convenient loop of host DNA. Latching on, the integrase positions the 3' hydroxyl groups over host DNA phosphorus atoms 5 base pairs apart. And splices in. Integration is completed by host repair mechanisms that fill the gaps with complementary base pairs. Host RNA polymerase transcribes the viral DNA into genome-length RNA molecules, some of which are further processed to become messenger RNA coding for viral proteins. The messenger RNA is threaded through a vast array of ribosomes to create the peptide chains that protease will cut to become viral proteins. Finding their way to staging zones under the cell surface, areas marked by GP120 proteins, these peptide chains encapsulate the viral RNA copies just arriving from the nucleus and provision them with protein tools essential to further replication. When aggregation is complete, the entire complement of viral RNA and proteins is corralled into a bud that breaks off the T-cell surface and floats into the plasma. Like an efficient nanomachine, the molecules in the immature variant assemble the capsid, ripening the virus to infect the next host cell.
Nice and dramatic music. All right. Okay, so let's talk a little bit about what happens. So along comes this virus, it gets into some T cells, gets into some dendritic cells, and it turns out that it's cytopathic, meaning it kills the cells. So over time, you start to lose some of these T cells. And in particular, there's different stages that we know that take place with the life cycle of this virus. So for example, upon the first exposure, normally you have something around 1,000 T cells, CD4 T cells per microliter in your blood. And that significantly drops upon infection. So within a few weeks, you start to develop these flu-like symptoms. That then is accompanied by this significant drop in CD4 T cells in your blood to levels below 500, which is kind of about the range where you start to feel sick and you start to become susceptible to infection. This is preceded by this stage that's essentially what we call the asymptomatic stage, where you start to recover some of your blood cells, your CD4 blood cells start to increase. So now you have a bunch of helper cells, not quite back to the normal level, but still enough to keep you protected. And so the result of that is that you have you essentially don't have any symptoms at this point. And this phase is quite variable depending on the individual, the environment, and can last somewhere between less than a year all the way out to 10 years. And again, most of what we're talking about today is without the antiretroviral therapy. We'll talk about that at the end. So this asymptomatic phase can last now much longer through drug treatments. But basically, this variable phase then is preceded by something called the symptomatic phase, where the counts start to get below 500. And at this point, you are susceptible to a lot of these diseases, pneumonia, other kinds of things that you're sick, but you still have enough CD4 T cells to keep you alive. And then within a short period after this symptomatic phase, then this develops into what's called AIDS, where your cell counts, your CD4 positive T cell counts drops below 200 per microliter in your blood. And at that point, you essentially don't have an immune system. You still have adaptive or innate immunity, but your adaptive immune system is essentially shot at that point. And the result of that is that at this point, because of the fact that you're essentially completely immunodeficient, that can then result in death. Okay. So what's actually happening? What are the features between these CD4 positive cells and the virus that allows all this to take place? So first off, as we learned a little bit, this HIV virus, when it binds to a cell that's expressing CD4, it's not sufficient to simply have this CD4 there. You need to have a co-receptor. And in this case, the co-receptor is either CCR5, so these are both chemokine receptors, CCR5 or CXCR4. Okay, and the CCR5 is normally expressed on CD4 cells, on dendritic cells, and macrophages. And CXCR4 is expressed on CD4 T cells, but you don't find it on these macrophages. So that tends to favor infection of these CD4 cells. And I should say that depending on the sort of co-receptors that are bound, you have different types of viruses. So some that prefer binding to this CCR5, which are called R5 viruses, and then some that bind to the CXCR4, which are tropic for, that are called the R4 viruses. Okay, which I just said here. So basically, and then there's a subtype of the virus that can bind both to CCR5 and CXCR4, and these are called dual tropic viruses, which basically 3 different strains of these viruses, even though they have a similar overall ability to infect CD4 positive cells, there are different tropisms based on the sort of host cells that they can get into. And I should note that we know that these co-receptors are really important because there's a group of people, actually this is a reasonably common polymorphism in CCR5 in Caucasians, where They don't really have very functional CCR5 on the surface of the cell. And the result of that is that these individuals don't tend to get infected. So even if they may have multiple exposures because of their profession or whatever, the result of that is that they don't get a productive infection. And so we know that this co-receptor turns out to be really important. I guess one question that I would ask is if they don't have CCR5, which of course protects them from HIV, what else do you think might happen to these folks? Yeah.
Their ability to activate other white blood cells are diminished.
Right, yeah. I mean, CCR5 is really important for getting activated T cells to certain places like we talked about before with respect to gut immunology and so forth. So they may have a little bit of a defect in terms of their overall immunity, but under these situations, they're actually favored. So you tend to find what we call long-term non-progressors, which are folks that have been in an environment or a situation where they should be highly exposed to HIV, and yet they don't develop an infection. And the reason for that is that they just have this defect in R5. So even if their immune system is somewhat depressed a little bit, and they may have some problems, under these situations, they're actually better off. Any questions? All right. All right, so let's talk a little bit about the host response and replication. So basically, infection occurs in various differentiated cell types, and we talked about macrophages. That's also true for circulating monocytes that haven't committed yet to the macrophage phenotype. This also happens in the brain, in cells that are called microglia, which are essentially cells that are kind of like macrophages. They're part of the innate immune response inside of the brain. And as a result, when the disease ultimately leads to AIDS, a lot of times this can be associated with dementias because there's considerable damage inside of the brain. And all of these, and this can also occur in dendritic cells, all of which are bone marrow derived. And all of these cells, infection in all of these cells plays a role in the pathogenesis. I just described, for example, what happens with infection in microglia. Obviously, when you have infection in your CD4 cells, that's going to depress your immune response. And so there's a significant component to that as well. So that's important to that. In addition, these DCs, as we'll see, can bind, sequester, conserve, and as well, they can actually disseminate the disease. So they can take virus from one place in the body, bring it into a lymph node, for example, and expose that to T cells, resulting in the uptake of that virus into the CD4 T cells. As well, DCs express CCR5, although it's at low levels, which is why we tend to find more infection in the activated CD4 population. All right, so let's take a look at the transmission portal of entry and the spread of the virus. Where is this all taking place? So this is an example where the virus might come in and through, in this case, cervical vaginal epithelium, makes its way across through that. Perhaps there might be a sore or something like that, gets past that barrier, and once it does that, then it's now present inside the lamina propria. And we talked quite a bit about the lamina propria inside the gut, but this is similar sort of mucosal tissue in other places as well. So the consequence of that is the virus can come along, it can get taken up into a dendritic cell. That dendritic cell can then traffic that to a place where you have essentially resting T cells, and those cells can then become infected, and then the result of that is that they may traffic into a nearby lymph node, where they then can expose that to other T cells, and then you start to essentially broadcast the virus. So now you have this virus being presented to other T cells. It's also being taken up into macrophages and so forth, and then they're trafficking around, interacting with other viruses, and eventually, over time, you start to become systemically infected. So the individual that is infected in one site in the body, you have a localized infection, that can then make its way into lymph nodes, into the lymphatic system, through the blood, and then it can make its way throughout the body. So there are essentially three different ways in which the virus can be transmitted. The first and most prevalent, over 90% of the infections occur through sexual contact. The second is through mother to child, through breastfeeding or possibly through the placenta, the bloodstream, although that's more rare. And then the third is through blood and blood products. And this is particularly true for hemophiliacs. In the early part of the disease, when it was first being discovered, one significant component, one group that was particularly sensitive to this were hemophiliacs, and they were getting that through blood products. So these folks are always going to the doc and trying to get blood products, and at that time, they didn't know what the virus was. They didn't know what was causing this disease. And so a significant number of people developed AIDS because of the fact that they're getting these blood products that are infected with HIV. Now, one thing that's kind of has been very important to understanding how this disease works is the use of something called SIV. This is simian immunodeficiency virus. So this can infect rhesus monkeys. And you can't use HIV. There's a different host range, and you can't take HIV and infect mice or Drosophila or whatever. The result of that is that they're not productive infections. But using SIV, which is very similar overall to HIV in terms of the structure of the virus, the genome, and the ability to infect CD4 positive T cells, it's been possible to really understand a lot more about the kinetics of this particular disease. So one example of this is using SIV for infection of these monkeys, and the result of that is that you can detect virus inside of this submucosal lymphoid tissue, and this is primarily taking place inside of these resting CD4 T cells. And then within a few days, as I described, then you start to see this virus is starting to spread to other lymph nodes. So it's starting to be disseminated throughout the lymphatic system. So what's happening? I mean, I showed you this slide where it takes many years in some cases for this disease to progress. So it's basically an infection. The virus essentially becomes quiescent, so it's not really causing a whole lot of problems. And then at some point that's down the road, in some cases somewhere between 1 and 20 years, you start to have this reoccurrence of this virus. And we call that a viremia. So there's an initial viremia that precedes the point of contact. So once a person is infected, they develop a viremia, the virus levels go sky high, and then the immune system is effective and is able to control that, goes into that non-symptomatic phase, which is somewhere between 1 to 20 years, and then enters into this clinical stage where the CD4 counts drop below 200, and then that can then result in clinical AIDS, which then somewhere between 1 to 4 years before those folks will die. And as I mentioned, there's essentially a flu-like disease or syndrome that's sort of like mononucleosis, and that is basically right during this peak viremia. This is when this is following first infection, which is also associated with a significant drop in these CD4 cells in the blood. And then the immune system controls it. So we always think about the immune system being shut off by HIV, but in fact that's not true. So part of the reason why you have this variable 1 to 20 region of non-symptomatic or asymptomatic stage is because of the fact that the immune system is actually able to control this infection and to maintain levels of virus at low levels so that the immune system is still functional. And I should point out as well that once you've cleared the viremia, so the virus levels drop, this then starts to stabilize. So you can use a certain test called an ELISA test or a PCR test to check for levels of virus that's present in the body. And at some point, it starts to stabilize after that initial infection. So somewhere about 4 to 6 months, as I mentioned, there's this drop, which is in CD4 cells, an increase in the viral particles. But then that starts to reach a set point, and that set point is basically then partly going to dictate how long that particular individual is going to be non-progressing to the next stage, which is symptomatic. All right, so that kind of looks like this, where basically there's this primary infection, and so we're plotting here two things. This is the viral RNA, and this can be measured again. In this case, this will be a PCR test. And so you can see within very, very quickly after infection, there's this significant increase in the amount of viral RNA that kind of peaks out, and then it drops to a point where it starts to stabilize about 4 to 6 months out, and then it's essentially plateaued until later in the response when the immune system can no longer seem to control it. So here you have the CD4 counts, which again, you have this rapid drop in CD4 positive cells in the blood, and then it starts to stabilize. But you see that it never fully stabilizes. Even when the RNA seems to be controlled, you see that the CD4 counts seem to drop over time. And then eventually they drop below 200, and it's that point where there seems to be this, once the cell count drops below 200, there doesn't seem to be very good control anymore of the virus. And so you develop a second viremia here out, which basically corresponds to what we call AIDS. All right, any questions about the kinetics of this or the overall response? All right. Now, one thing that's important is to note that, there are different routes of infection. We talked about different ways that the virus can get into the body. One particular place we've talked about before is in this GALT, this gastrointestinal associated lymphoid tissue or gut associated lymphoid tissue, where, as you guys know, there's a lot of T cells there, and those T cells are very important in protecting us from nasty stuff that gets into our gut. And in this case, remember back, those cells there are actually constantly getting activated. So even though it's sort of usually a tolerant environment, there's still a lot of activated T cells that are making their way out of the lymph nodes and Peyer's patches and so forth, and making their way back into these areas, these GALT areas. And in this case, certain naive T cells, which are CCR5 negative, and then memory cells, which are going to be CCR5 positive, which is important for getting them into the certain places, they're going to be susceptible to this virus infection. So here's just an example of what you might see in terms of the numbers of these cells inside the gut. And I'll show you a movie about this in a little bit. Basically, if you look inside the gut, and you can see this precipitous drop in the number of these CD4 T cells in the gut, that also corresponds to an increase in the number of in this case, SIV RNA molecules. So we're looking at a monkey here, which then drops. And then over time, you don't see that reemergence of this disease, because we're only looking at this for about a month. But the important point is that those cells drop. And in fact, it's a very much more profound effect in these cells in the gut, because they're constantly getting activated. And as I'll show you in a second, or what we were talking about with respect to kappa B activation, these are cells, because they're constantly getting activated, they're not going to have a long latency phase. So once they get infected, they tend to convert that and activate viral replication. And they tend to spread it pretty quickly. So you tend to, through this route of transmission, as opposed to getting it through the blood or something, this route of transmission tends to lead to very, very quick depletion of these CD4 cells. Now, you probably wonder, that latency phase, why is it that the immune system is capable of controlling this if you're killing off a bunch of your CD4 cells? Well, obviously, one is because you're producing a bunch of antibodies, and there's a variety of different antibodies. And in fact, one of the tests for HIV is an ELISA test, which looks for antibodies generated against the virus. Okay, so that's one thing. And those antiviral antibodies turn out to be very efficient, at least early on in the disease. But you also have a component of the antiviral response through CD8 cells. And again, here we're looking at SIV infection, but this is also true in humans infected with HIV. So here you can see a sort of an experiment that was done, again, using monkeys infected with SIV. And basically, we're looking at a monkey here that is basically just a normal monkey. It's our control. And we're looking at the amount of virus using a PCR test for viral RNA. And you can see that, after immunosuppression, there's this, basically a slight increase. But if you get rid of the CD8 cells, so they have basically no CD8 cells, in a different monkey, you can see that the response, the viremia, is much higher than it is in this control monkey. So that tells us that the CD8s are actually playing a really important role in trying to control this virus, and presumably they're using cytotoxic mechanisms to kill off CD4 positive cells T cells, dendritic cells, and what have you to try to limit the spread of this infection. Okay, so you guys have probably heard of this antiretroviral therapy, which is basically combination therapy. And the reason for this, so basically what these drugs do is they tend to kill off viruses. So they can block the replication machinery, they can block the entry of these virions into cells, and they can block that polyprotein proteases. So by using certain protease inhibitors, you can prevent the processing of these polyproteins such that you don't generate the GP41 and GP120 that are required for infection. And so if you look within days, there's a very, very quick response. Within about a day or so, you lose essentially 2 log in this viremia, so that it's very effective. And then over time, it drops quite a bit, although it starts to plateau. So there's a certain point where this antiretroviral therapy is used where it's clearly gotten rid of most of the virus, but it does not cure the HIV infection. And we know this because if monkeys or AIDS patients or people that have HIV go off of this therapy, they very quickly, their viremia is reinitiated in these folks. And so we know that these drugs are very effective, but they don't cure the HIV infection. There's always going to be some of this latent provirus that's present in cells. Any questions about that? Yeah.
In the previous slide, so the control was the green.
Yes.
Why is the viral DNA lower in the one that had depressed CD8?
Why is the viral RNA lower? Are you saying like here or something like that?
So after the depressed CD8 it's replaced, right? So they depress it for a while.
Correct. So why does it come back up? I don't know. That's a good question. I'm not sure. Any other questions? Okay. So as I was mentioning, you have this immune response that's important for controlling this initial viremia. So again, within the first few weeks, here we're looking at the virus inside of the plasma. And you can see this big peak, which then can, for a variable period of time, stays relatively low, although you can see that it's kind of cycling. So there's a little bit of a cycling in terms of the level of virus that's in the plasma in this individual. But if you look at some of the immune parameters, for example, here you have HIV-specific CD8 positive cytotoxic cells. And so within the first couple of weeks, you start to get this CD8 response, which we know is very important in controlling the virus. And you also start to generate antibodies, one to the envelope protein and then another to the core protein, which are also key in regulating the response of this. So very important in controlling this. One thing that's kind of interesting, though, is that we know that the virus can change over time. And we know that because some of the original studies trying to develop antiretroviral therapies, the virus over time would kind of change so that it was no longer susceptible to these drugs. And so the result of that is that with this HAART approach, the point is this is a combination therapy. So you're not just targeting the retrovirus. You're not just targeting the polymerase, for example. You're not just targeting the protease. By using these different approaches, you're basically trying to throw everything at this virus at once to try to achieve this huge drop in the amount of virus and prevent it from mutating. But oftentimes, these viruses are very good at mutating, and so the result of that is that, over time, there is some concern that some of these viruses may develop resistance, and so that's why the drug companies are really interested in continuing to improve these and develop different approaches. The other thing that's important about that is that you can see that you have these antibodies that are developed against the envelope and the core protein. But if these are changing, these are also changing. This virus, like all viruses, likes to mutate. There's a lot of error-prone replication that takes place that gives rise to variants in the population. And so the consequence of that is that antibodies that are generated, say, early on in the response may no longer be effective because there's a subpopulation of virus that comes along and kind of changes its spots, right? We talked about that a little bit last week when we were talking about trypanosomes, where they have those variable surface glycoproteins that can change over time. In that case, it's not mutation, that's because it's of gene conversion, but it's really the same overall concept that as these populations of viruses are changing, the immune response has to be renewed now against those new epitopes. Any question? Somebody raised their hand? Okay. Now, one other issue that is involved in this, and I mentioned this early on, is that a lot of the early folks that were in the population that were getting infected with HIV had some unusual symptoms that they were coming along with. So, for example, the significant drop in CD8 T cells, and also things like fever and fatigue, lymphadenopathy, GI tract symptoms, oral candidiasis. So these are things that basically you might find in somebody who's immunodeficient, and at the time it didn't make a lot of sense, but now we know that these are really a consequence of just not having an effective adaptive immune response. People that progress to AIDS also happen to have a lot of much enhanced susceptibility to parasite infection, protozoa, fungus, bacteria, and viruses. They also have an increased risk of neoplasms. And examples of that are Burkitt's lymphoma, cervical carcinomas, and Kaposi sarcoma. So these are all examples of where a normal immune system would be able to recognize that these cells are somehow changing. They have mutations that are propagated. Normally those immune cells, the T cells, would be able to wipe those neoplastic or transformed cells out, but because the immune system is not functional, they can't do so. All right. So why is this? How are the cells eliminated in this case? One example of this is that the CD4 cells are basically getting killed. So the virus is itself cytopathic. And then on top of that, there is this CTL-mediated killing. So these CD4 cells are infected. You're going to have some CD8 cells that come along, can recognize that, and then can lead to killing of those cells. And the last thing I wanted to show a brief movie that kind of nicely describes this process.
The human gastrointestinal tract, also known as the GI tract, is vast in surface area to maximize the absorption of nutrients. Most of the body's immune cells are contained in the GI tract, making it one of the largest immune organs in the human body. One important subset of immune cells is known as the CD8 T cells. These are T helper cells that provide protection to the gut against invasive bacteria or naturally occurring gut flora. HIV is a retrovirus that causes fatal immune deficiency and needs to infect human cells for its own replication. In the body, HIV preferentially infects activated CD8 T cells. This is because HIV must bind to CD8 to gain entrance into the cell, as well as to a co-receptor called CCR5 that is expressed in activated T cells. Due to bacteria normally found in the gut, the vast number of gastrointestinal CD8 T cells are constantly activated, making them especially susceptible to HIV infection due to the high level of CCR5 expression on the surface. Approximately 2/3 of the CD8 T cells in the gut are immediately lost within weeks of initial HIV infection. Compared to the peripheral blood, which contains only a fraction of the body's total T cells, the initial loss of CD8 T cells in the gut is far more profound. With so many T cells lost during early HIV infection, the normal barrier formed by the gut lining is severely compromised, allowing gut bacteria to cross the gut lining into the bloodstream in a process known as bacterial translocation. Bacterial translocation leads to immune activation in the body. and this inflammation can be damaging to the immune system in the long term, causing both heart and neurological diseases. Immune activation also makes remaining T cells even more prone to HIV destruction. Today's HIV treatment, known as antiretroviral therapy, or ART, is recommended to patients before CD4 counts drop dangerously low, or when AIDS-defining illnesses occur. In the peripheral blood, ART can reduce viral load to undetectable levels and often restores normal blood CD4 counts. However, its effect on gut CD4 T cells is much more delayed and incomplete. Reconstitution of gut CD4 T cells may take many years. This may be one reason why heart and neurological diseases remain a problem for HIV-infected individuals, despite antiretroviral therapy.
All right, we're going to stop it there, but I'll put that on the website for time. One other thing is, if you submitted a midterm for regrade, I have those here, so,
Also,
stop. Okay, anyway, so we'll see you guys on Wednesday.